Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show. Today, bringing another really awesome guest involved in science and technology, creating a better tomorrow for many people out there. Today on the show, we have the honor of being joined by Dr. Julia Moore-Vogel, who is assistant professor and senior program director at the Scripps Research Institute, where she is responsible for managing a broad portfolio of patient-centric health research studies, including some very interesting work on long COVID, known as the Lockett or long COVID treatment trial, which is this really interesting sort of fully remote randomized placebo-controlled trial involved with individuals with long COVID and testing whether GLP-1 drugs can reduce or alleviate symptoms, and really interesting repurposing story there as well. Prior to this current role, Dr. Vogel managed the Participant Center for the All of Us research program there at Scripps. And this is the NIH program that we profiled a little while ago, focused on, studying why some people get sick or stay healthy. And she was charged with recruiting and retaining somewhere 350,000 different individuals to represent the diversity of the United States. So really interesting to hear about their role in that program as well. Prior to joining Scripps, Dr. Vogel created, proposed, fundraised for, and implemented various research and clinical genomics initiatives at the New York Genome Center and Rockefeller University, overseeing proposals, execution of grants, including a $44 million NIH Center for Common Disease Genomics in collaboration with 20 other scientific contributors across 7 institutions. She also managed corporate partnerships, including a really cool one with IBM, assessing the value of several genomic assays for cancer patients. Dr. Vogel has an undergraduate degree in mathematics from Rensselaer Polytechnic, went on to do a PhD in Computational Biology and Medicine at Cornell, and also did her MBA from Cornell. And a lot of really cool topics to get into today. Really honored to have him with us. Dr. Julia Moore-Vogel, thank you so much for taking time to come on the show.
Thanks for having me. I haven't had that much of my CV read in a while. It's fun to hear.
No, you've done a lot. You've done a lot, and I'm gonna really enjoy going through a lot of this with you. But love to start off at the very beginning, because what I didn't mention in your bio is that you were in doing your PhD, you were involved in this really interesting tri-institutional program in comp bio and medicine that brought together not only Cornell, but also Memorial Sloan Kettering, Rockefeller University. And you studied, I know we're going to be getting into long COVID and other genetic diseases, but you studied the intricacies of embryogenesis and morphogenesis and doing all sorts of interesting single cell imaging. So stuff is really hot nowadays in 2025, but you were doing it 10 years ago. Talk a little bit about this and how some of these topics sort of became sort of the crux of your PhD.
Yeah, absolutely. I loved math throughout my life, and I decided to study it in undergrad. And I really didn't know much about research or that you could get a PhD at the beginning of my undergrad. I grew up in this really rural area where very few people went to four-year colleges, and I wasn't exposed to a lot of the type of work that I do now. And so I basically, after college, said, okay, more education is better than less, and they'll pay you to get a PhD, whereas you have to pay to get a master's program. So this seems like a good idea. I was like super naive going into it. And when it came time to choose a topic in a research lab, I was really thinking about what is going to be the most in-demand skill set that I could build. And so I was really interested in mathematical modeling. I think it's very exciting to be able to use math to explain the things that we see in the world and then perturb the parameters and look at what does the model show us, being able to sort of predict things that we can't see in the real world. I still love that sort of work. But when I was thinking about what's going to be the most employable, I thought, okay, large scale data analysis is where the field is going. And so that was how I ended up in the lab looking at embryogenesis, where what we did was a 3D scan of this worm called C. elegans, which most people don't know, but it has won many Nobel Prizes. And one of the really great things about it is it has an invariant lineage. So it has the same number of cells every time that arise in the same way. So what we were doing was characterizing the statistics of each cell. What is the normal, amount of time that the cell is there, the normal, time that it divides, what genes it expresses, that sort of things. And then we characterize normal, and then we would perturb it with RNA interference, one RNA, one gene at a time, basically. And so the idea was to sort of understand what's the normal development versus what's going wrong, and do that in a really rigorous mathematical way. Um, it is it really prepared me well in some ways for this moment because data analysis is so important in everything that we do. And so there's a lot of skills that I still use, not everything, but I still am really glad that I did that. And the program that I was a part of is still going and excellent. It's really cool to be able to choose between institutions and to have so what we do in that program is you go to Ithaca for a year to do your classes. And so you have the educational opportunities of this amazing, large Ivy League university. And then you can choose to do your research there or at the more medical focused campuses in New York City. So Sloan Kettering and Weill Cornell.
Hmm.
And so I ended up at Sloan Kettering actually. So my degree is from Cornell, but I actually spent most of my PhD time at Sloan Kettering. And it was a really nice program that I definitely recommend to people that are looking into computational biology.
Yeah. Yeah, I was reading all about it. I didn't know it existed, and I just thought, wow, I would love to do something like this. But
Yeah, totally. There's
yeah,
like 10 students a year, so it's pretty
yeah,
small, but
yeah.
very, very cool and very supportive of your individual research aims.
Yeah. And I enjoy it. Again, the topics of embryogenesis and morphogenesis are just so fascinating, not just for development, but just the connections to all things, how cancer develops and regenerative medicine and everything, systems biology. And it's so I was excited just reading that beginning. So,
Absolutely.
but yeah, so from there you move on to get your MBA, also Cornell, and then you head off to the New York Genome Center, and here various institutes and investigators and clinicians then you start sending you samples to do all sorts of analysis in terms of sequencing, computational analysis. You publish on everything at this point from the genetics of nasty tumors like glioblastoma. You did this really cool sort of pharmacogenetic work in children with asthma, looking at albuterol, which kind of interesting because we just did an episode a couple of weeks ago on the sort of the pharmacodynamics of albuterol that 50
Cool.
years later that has been on the market. We're still learning things about it. Talk a little bit about this. I mean, because clearly, the things that you were doing then, obviously learning new things, but as we'll be getting into repurposing and learning about new uses for drugs, finding interesting things for things that have been around 50, 75 years as well.
Yeah, so the through line between those two things was I went into the computational biology program because I liked the idea that you were working at the interface of math and biology, and that you could kind of bridge the two languages and use tools from each to solve problems in ways that people hadn't thought about before. And I sort of had the same idea for my role at New York Genome Center, which was bridging the business world that I learned about in my MBA and the science world that, obviously is so crucial to the work that's done there. And so that was a real crash course for me in so many different things. You mentioned the different institutions. So there are really excellent institutions all around New York City that put in seed money to build this organization that I was part of. And so you had a lot of really high-powered stakeholders coming together with different ideas of what needed to be done, and we were trying to do what was best for the institutions all around the city as a whole. So there was a lot of different points that needed to be addressed and different stakeholders to work
Mhm.
with. I ended up after a couple years there becoming the right-hand person to the CEO at the time. And that was a really interesting role because whatever would come to the CEO's desk would come to mine. So the types of things that I normally would not have exposure to until later in my career, I was able to, have a front row seat to, like writing the grant that you mentioned at the beginning, the Center for Common Disease Genomics grant. I was really orchestrating that so that my boss could focus right on writing the scientific pieces. I was orchestrating the 20 collaborators and all of the people at our institute and trying to, sort of bring everything together in a cohesive way. And it was really exciting to get that grant because we were a new center and it really wasn't expected that we were going to get it. So it was a great victory to have. That really prepared me for all the grants I've written since. That was really the first one that I was deeply involved with, and it was an excellent learning experience in many ways.
Very cool. So, you head on to Scripps, and as I mentioned in your bio, one of your previous roles is in managing the participant center, and we done a few episodes. We had
Yeah.
Josh Denny on talking about all of us, and then Rachel Romani over the Veterans Administration with their sister program, Million Veterans. When it comes to again, in this case recruiting hundreds of thousands of individuals, and obviously because you're there and you have not just the Scripps, but there's also the digital trial center that you're involved in. Interesting stories, learnings about this, because clearly all of us has been, it's been around for a couple of years now. You clearly had a major role in making sort of this program available to who participants want to be involved in it. And we'll be talking about digital recruiting and all that in a little bit when we get to the Lockett trial. But talk a little about this as well, because I think it's a very important piece of your responsibilities prior to what you're doing now. And if we can just sort of reintroduce sort of Scripps' role in
Yeah.
all of us as well, that'd be cool.
Absolutely. So I remember when they announced the All of Us research program, I was still working at the New York Genome Center, and I just assumed that we would apply our way in, as we do for other programs. And I ended up switching institutions, and I was so thrilled to get to a place where I didn't have to write the grant. The money was already there. And so it was really Steve Steinhubel and Eric Topol and Katie Bakamoto who got the program started. And so, I was there to help with the implementation, and it was really exciting to have the vision laid out that anyone anywhere around the country can join us, and we just need to figure out how to make it accessible for them. And that's what we've been doing, throughout my time at Scripps. Um, there's a lot of, I think, misunderstanding about why participants are historically underrepresented in biomedical research, especially folks who are not white. We say underrepresented in biomedical research based on race and ethnicity, or UBR race ethnicity. Um, you know, there's a lot of misconceptions about digital access, for example. Now, there are people, of course, that are UBR race ethnicity that don't have digital access, but the large majority do. And so, what we have been trying to do is help people understand, okay, there are lots of people with digital access that are UBR race ethnicity that we can reach through these digital campaigns and, all different online ways. And then there are folks that really don't have access that you need to be, engaging with in person or in a more high touch way. But that's also true for all the rural populations in the US, all of the people with low educational attainment that maybe are not able to engage with the material or really need to talk to someone before they build a level of trust to be able to share their information that is so personalized. So we've talked a lot about the different things that need to be done to reach the program's goals and how we can reach them efficiently. And it's been really interesting to watch, folks sort of learn from us and us learn from others as well. One of the best things that I've learned in the course of the All of Us research program is from the communities of color that are represented on the leadership boards throughout the program, and hearing directly from different communities about the hesitations they hear from their constituents and the education that they want and the return of value that they want. All of those things have been really great learning experiences for me. And we are still involved in All of Us today. Our role is a bit smaller, but we are still doing a lot of the digital work. We were just talking an hour ago about analyzing the data from a remote blood collection pilot, which was really cool. So we sent these little at-home micro needle collection devices that anyone can use. You just put it on your arm and press a button. And we did a pediatric pilot. And so my child was in it. She was 7 at the time. I had a cousin who put it on her, 6-month-old or whatever. And, there's not a great way to collect blood from pediatric participants in the program yet, or collect DNA from pediatric participants yet. And this was a pilot that we were doing to be able to enable broad recruitment of pediatric participants at a reasonable cost. And so we're still doing those sorts of projects, and it's still really fun.
Really cool. So, that leads us into the theme of long COVID, and I guess before we get into um, a lot of the science that you've published, and you did this really comprehensive sort of Nature review in microbiology back in 2023. Would you mind just saying a couple words about your story? Because I mean, I've seen it written up, and prior before we get into everything going on, would you mind just saying a couple words about your own long COVID experiences? And I mean,
Yeah.
I guess everyone has one. I know there's one in my family. My brother-in-law would come on and talk all about it, but would love to
Yeah.
hear about what you experienced in your own day to day, and then we'll.
Yeah, it's I'll just tell you at a meta level before I start with me, it's amazing how many people are affected by it and how few people want to talk about it because it's so stigmatized. Being really tired, it's just it's very frustrating how it's perceived as a moral failing by a lot of people. And so I feel kind of an obligation to talk about it because I feel like I'm in a position of privilege where my employer understands and is not going to count it against me. So yeah, I have had long COVID and myalgic encephalomyelitis, MECFS. Some people call it chronic fatigue syndrome since July 2020. And I had read about long COVID before I got sick, and we had been very conservative about going out and following all of the restrictions. But we lived in an apartment building with a shared indoor hallway at the time. And so I would take my daughter out to play. She was 2 at the time. That was the only time I ever left the apartment, was just to take her out to get some fresh air. And honestly, I was reluctant to even do that because I was worried about COVID, but my pediatrician told me I needed to. So I agreed. And my guess is that's where I got it. Like we wore cloth masks in the hall thinking that was enough at the time because that was back when they were saying don't buy the N95s, leave them for the medical staff. So it was not enough for me. And I was very, very sick. I took 3 weeks off of work. And I had trouble breathing, but was able to, with medical management, avoid being hospitalized, which was good. Um, and I just never got back to my prior baseline. And it was something that I had an eye on the whole time because I had read Fiona Lowenstein's New York Times article, in April 2020 or so. And I think maybe some of Ed Young's pieces had come out by that time too. But I was watching out for it. And I was sort of like, okay, if I get to the 4 week mark, maybe we should start worrying. And then, it's around 6 weeks, I started joining the online groups to learn from people about long COVID. And at that time, there were people from March who were starting to recover. And so I was thinking, okay, I am going to make it my goal to recover by the end of this calendar year. And, by December 31st, I'm going to have my glass of champagne and be toasting my good health. And it was incredibly naive of me to think it was within my control. It's totally out of my control, right? This is a chronic illness that people have been suffering from for decades, MECFS, I mean. And it has been humbling in every way you can imagine. We ended up last year moving back in with my parents. I'm at my parents' house now, and I was really lucky to be able to be supported to keep my current job while I did that because I needed more help around the house to be able to, do things like I can't change my sheets without inducing post-exertional malaise, for example. So, my mom and I have a schedule where she comes in and changes my sheets. Things that I would never think about as being tiring are now major barriers in my life. So it has changed my life in every conceivable way, I would say. And I'm happy to share more, whatever you think would be of interest to the audience.
And I appreciate you sharing all that because I also read that you are a long distance runner, prior to
Yes.
all this as well. So I
Yes.
said somewhere like 90% of your, lung capacity, it was wow. I mean, it was just really night and day. And I mean,
Yes.
I've again in my own experiences with my brother-in-law and others,
Yeah.
it was rather a striking story when I was reading about it.
Yeah, the other thing I'll tell you about it is, well, I'll tell you one thing in terms of the night and day is I used to be a person that was productive every second of every day. Like if I was sitting on the floor and there were toys out, I would be organizing the toys and putting them away. And I remember when I first got sick, my husband was like, "Wow, you're not putting the toys away. I guess you really are feeling really sick." Like that was a major red flag. And now it's like I can't remember the last time I put away the toys. Like I never have the energy for that. Um, and then the other thing I wanted to mention about it is I have worsened every year since. I started out mild with mild ME, I would call it, where I couldn't exercise, but I could still walk for a mile, and that was okay. And now I am on the high end of moderate, trending towards severe, I would say.
Mm.
And in the beginning, I got worse because I didn't manage my symptoms well enough and I kept overdoing it and not realizing I was overdoing it. And I didn't recognize post-exertional malaise for what it was. I didn't know that it could be triggered by just cognitive tasks. I thought of exertion as physical, but
Yeah.
actually just sitting at my computer, reading or even just laying down thinking can trigger post-exertional malaise. So it took me a long time to understand my limitations enough. And that was what triggered the move here, that we were just like, okay, for me to be able to avoid triggering post-exertional malaise and stop the decline, we need more help. And then, I was doing well in terms of like maintaining the plateau. And then I got reinfected in January,
Mm.
which I work very hard to avoid COVID. I hardly go anywhere. I mask, pretty much anytime I'm with people that are not part of my immediate household. But my daughter is in elementary school and she brought it home from school. No fault of hers. I'm not blaming her in any way, but it's just very hard to avoid in society these days, no matter how many precautions you take. And that, made a substantial dent in my baseline, and I've not recovered since then. And so, for the people out there that don't have long COVID, I would really encourage them to take special care around the people that do have long COVID or that are vulnerable to COVID in any other way, because a lot of people have sort of quote unquote moved on and they don't think about it anymore, but it's still very dangerous to a large segment of our society, and we should treat it that way.
Hmm. Absolutely. Absolutely. Well, let's again, before we get to the LACA trial, let's talk a little bit about some of your research in the ensuing
Yeah.
years, because I mean, the one thing that, and we'll put a link to this, the very comprehensive January 13, 2023 paper, you, Dr. Topol, others on your team, it's entitled Long COVID, Major Findings, Mechanisms, and Recommendations. And you really, you take the reader through really like 7 different sort of chapters of it, one being just the scale in the sense, tens of millions of people affected globally, into this principle that, we're not really dealing with the disease here. We're dealing with something more akin to a syndrome, and there's multi-organ involvement, neurological, cardiac, immune, metabolic, many different mechanistic pathways, different risk factors, of course. And then when it comes to the sort of the pharmaco or the genomic component here, very heterogeneous presentation, as you were just explaining. Take a little time to, again, with your own experience, and then obviously this very comprehensive deep dive, what we know about long COVID in 2025 in terms of its clinical presentation.
Yeah, it's actually amazing how much we know about long COVID, given that it's only been around for 5 years. There has
Right.
been so much high quality research done. And I remember when I was writing that article with the two other colleagues on there, Hannah Davis and Lisa McCorkle, are from the Patient-Led Research Collaborative. So they're also people with long COVID themselves. And I thought it was really cool of Eric to, partner with the three of us as patients to put this together. When we were putting this review together, we actually had more citations than the journal would allow. And it was heartbreaking to take out some of the things that we thought were really foundational, because there was just so much excellent literature already 2 years ago. And of course, it continues every single day. It feels like there's another really interesting paper about long COVID. So what we know is that there are over 200 symptoms that can be involved. We know that there are several different biological processes that are disrupted, many of which are mentioned in our review, but there's been even more, new data coming out with more about mitochondrial dysfunction, more about
Yep.
viral persistence. What we don't know yet is what pieces are causes and what pieces are effects. And so that's part of what we're still trying to learn through a variety of mechanistic studies and other things like that. We know that it is extremely common. The estimates are now about 400 million people worldwide have been affected by long COVID in some time or another, including 1% of US children, which is
Mm.
higher than I think a lot of people realize. In part because the symptoms can be kind of nonspecific, right? You're tired, you can't think as clearly. Um, you can have GI upset and many, many different things, but um, it can be hard for children to articulate it very clearly as a medical problem. And so a lot of times that makes it really difficult to identify in children. Um, so there are many different things that can go wrong. Two of the most common comorbidities that we have really built on the research of are ME/CFS, myalgic encephalomyelitis, chronic fatigue syndrome,
Hmm.
and different types of dysautonomias, in particular POTS, postural orthostatic tachycardia syndrome. And so both of those are present in about half of people with long COVID. Some people have both, some people have neither. But those are two, really fundamental comorbidities that we have learned a lot from in terms of management. And most of the treatments that we mentioned in that 2023 review are from the literature on POTS and ME/CFS. And now we are starting to come out with, more data about how they and other drugs can be used in long COVID.
Hmm.
One of the most important things is we do not have an FDA-approved treatment for long COVID. There are some things that we can do to lessen the symptom burden. The best one, in my opinion, is pacing, which is doing less. And we have another study on that we can talk about. But
Yeah.
there are some pharmacological and other support options, like, increasing salt intake for POTS and those sorts of things. So there's a lot that we can do to try to improve management. But there are still many, many people that are bedbound. Some are unable to speak, unable to tolerate regular food, those sorts of things. It can get to a very extreme level where the quality of life is so low that people are not even able to advocate for themselves, not even able to ask for the research funding because their restrictions are just so extreme.
Hmm. Hmm.
How's that for an
And,
overview? Did I miss any
no,
of the
no,
key points?
no, it's awesome, and it touches into the next year. It's September 13, 2024. You publish long-term changes in wearable sensor data in people with and without long COVID, and it's in this study, and I think this is a prospective study looking at something known as DETECT. Where
Yes.
you start the decentralized work with wearables and start drilling down on things like a resting heart rate, sleep quality, step count, learning about sort of the day to day. And I was wondering if you could also, because obviously this hits on the everything the Scripps is about in terms of
Hmm.
the decentralized, digitalized, distributed clinical trials. But talk a little bit, because I think this is a very nice paper in synergy with all the biological stuff that you were learning, and sort of goes hand in hand. So can you also talk about this one?
Yeah, absolutely. So that work was led by Jennifer Radin in our group, and it was using the DETECT data, which we originally started that study built off Jennifer's work in flu, which showed that if you look at sensor data, you can actually see that someone's resting heart rate is changing before they show symptoms of flu. And so the idea was like, okay, let's just have everybody share, as many people as we can, share their wearable data, since we know there's this COVID-19 pandemic and people are sitting at home wondering what they can do about it. They share their data, and then we can learn about, okay, what does it look like the couple days before they report their symptoms? And are there things that we can identify to be able to, trigger someone to take a test? Just, at that time, it was really hard to find tests. Now they're everywhere. But at that time, it was, trying to make optimal use of the resources that we had, but also, helping people know that they should rest, or, consider testing before they see a family member, that sort of thing. And so there were a few papers on that, mostly led by Giorgio Coir in our group, who did a lot of the analysis and is an AI machine learning scientist. And so they were able to find signals, in particular in resting heart rate increasing before the person tested positive. And we did some follow-up work on that. And then this long COVID study was sort of a secondary analysis out of that, right? Since we know many people tested positive and many people told us that, we were able to compare those who we later surveyed and said, do you have ongoing symptoms? Compare those that said, yes, I do have ongoing symptoms, and those that said, no, I don't have ongoing symptoms. And so we saw that those that said, yes, I do have ongoing symptoms had fewer steps per day than their pre-infection baseline, whereas those that didn't recovered to their pre-infection baseline. And so that, aligns well with our expectation, of course, about people being fatigued, but it was nice to have it validated in the data.
Awesome. Awesome. Okay, well this all leads now perfectly into to Locket, because this is a fascinating study that you've designed. Again, it's fully remote, randomized, placebo-controlled, and focusing on one of the GLP-1 drugs, Tirzepatide, and I believe this is going to be going on for 12 months,
Yep.
looking to see again how the use of and again, we've had the Lilly folks on and the Novo folks, and we've talked, we've done some GLP-1 shows, and it's just fascinating how many things that these drugs do. And again,
Yeah.
coming back to the principle of we're dealing with a syndrome here, and not, we're not dealing with diabetes or Alzheimer's disease. We're dealing with this basket. Could you talk a little bit about, talk about the background of who came up with this? Like, hey, this is an idea for the long COVID study, but when it comes to metabolism and anti-inflammatory effects and immune modulation and weight loss and neuroprotection, it seems like it's a natural fit to
Yeah.
what long COVID's all about. But walk us through a little of the history of
Yeah,
the
absolutely.
concept, if
So,
you will.
Eric and I have been trying to run a study like this for years. We put in some different applications that did not get funded, even before the GLP-1 drugs were, common knowledge. Not about GLP-1 drugs, about other drugs, because we just thought that, people with long COVID have such a hard time getting anywhere that it's a
Mhm.
perfect fit for a decentralized study to have, this group be able to receive the drug at home, improve accessibility for those that are housebound and bedbound. And of course, I wanted to do something about the illness that I have, and this just seemed like a great opportunity. Um, so it was really Eric's idea to try with a GLP-1 drug because of everything that you talked about. We know it's decreasing inflammation. We know there's inflammation in COVID and long COVID. In particular, neuroinflammation is one that we're really excited about. And so it was over a year ago that we, finally got the go-ahead from funding from the Schmidt Initiative for Long COVID, who's funding the
Mhm.
study. And, Lilly agreed to provide the drug and distribution. And so it's been a long road just to get to this point of launching, many years. But in terms of the drug, in the meantime, many stories have come out about people taking this drug with long COVID. So it started from people that were taking it for the original weight loss purposes and realized, hey, my long COVID symptoms are much better too. And then more and more providers started using it off label for that purpose. And one of our colleagues, David Kaufman, presented data at the NIH Recover TLC conference in September, I think, recent months, where he collated stories on over 350 individuals with long COVID and or myalgic encephalomyelitis, chronic fatigue syndrome, and 60 to 90% of them found that their symptoms improved on a GLP-1 drug. And it was different GLP-1 drugs, whatever the
Sure.
participant could access. But that was way better than I expected. I was hoping for like 20 to 30% improving just because of all the subtypes of long COVID and that we didn't really know which ones it was going to help. So as more data comes out, while we've been planning the trial, I'm feeling even more optimistic about the potential for this intervention to make a difference.
There is a long COVID treatment trial website. We will obviously put the link to that
Great.
in the show notes. But could you, Julie, could you just take a little, because again, there's a big frequently asked questions section, and again, there's pieces of this again being a completely sort of virtual study where devices, whether they're the blood testing, the study drug distribution, how samples are collected, all that. Could you sort of walk through what that's all about? Because obviously very different than, headed off to, my phase 1 center over here to do my dose escalation and in the traditional way, let's say.
That's
But
right.
take a little time in this one, please.
And in terms of the trial design, so we are really excited to be able to offer everything to people at home. The first thing that you do is engage with the My Data Helps, it's called app by our tech partner, Carevolution. So it's the same partner we've used for the All of Us Research Program, for Detect, for many other studies that we've worked on. And you can go through the screening process there where you take the eligibility survey, you upload some information about your diagnosis, and our team reviews that and then lets you know if you're eligible or not. And I should say the team is available throughout the process for questions that people have, and they're answering many, many questions. So please be patient with them. But we have a few different RNs and others that are answering questions. RNs for the more clinical things and the medical administration type work. But we're really there to support you throughout. So you do your survey, your eligibility survey, you get invited to the study, and then you go through the consent process. So again, you can review the material on your own or ask, any questions that you have to the team. Then you do baseline surveys. And after that point, you're invited to order both the Garmin wrist-worn wearable for tracking things like sleep, heart rate, and physical activity, and then also a smart scale. So we're asking people to take their weight every week. And that's to make sure that folks are in a safe weight range for them because we know that the drug was originally for weight loss and we don't want people losing too much weight.
Mm-hmm.
So MDs will be communicating with folks about their weight throughout as needed. And then you can order the study drug. So half of the people will be getting placebo, half of the people will be getting medication. And since it's double blind, we won't know who's on medication or placebo as well throughout the trial. And so an MD works with each person individually on their dosing based on what they tell us on a weekly and monthly basis. And the MDs the whole time will not know if you're taking the drug or the placebo. And so I'm really interested to see, how that works out and what happens with weight loss for people on drug and placebo and such. Um, Anytime you have a side effect or some, a medical concern, you can write to the study team. We also have a 24/7 phone line. But of course, if anything is a medical emergency, people need to just call 911 and go.
Hmm.
Um, for the biosample component, so 50 participants will take their own biosamples with the at-home microneedle device that I mentioned we were trialing in all of us at 3 different time points. And so we'll be doing some clinical labs with that. We'll be doing proteomic analysis to look at all the proteins, all at baseline, at 6 months, and at 12 months. So we will be comparing the differences between the people on the placebo and the people on the drug, who both will be taking biosamples to see if there's any signatures of the drug's effects, but also to look at subgroups of people that do and don't respond to the drug, assuming not everyone responds, to try to see if we can understand the mechanism behind what the drug is doing as well. And I want to pause there for a second because you look like you want to ask a question.
No, no, keep going. I'm
Okay.
really enjoying
All
this.
right. So, we're starting people on the standard dosing for the drug because that's what's FDA approved,
Okay.
and we are sort of building on the initial approval. There have been people doing microdosing and different things that are not, currently within the label, and so we're sticking with the label, and microdosing is sort of a future aim. And people will stay on each level of the drug for a month before they consider increasing. And so it's really a joint decision between the study MD and the participant about increasing based on how they're feeling, what kind of side effects they're having, and that's that sort of information. So if people get up to the highest dose, which is 15 milligrams for an injection
Hmm.
once a week, then that will take 6 months because there's that many steps to get up to 15. So that's why it's a 12-month duration study so that we can have
Right.
folks at that maintenance dose for a substantial amount of time. I should mention also the first injection is done on the phone or video with one of the study nurses because we really want people to have clinical support to make sure that they're doing that correctly. We welcome people to have caregivers involved in any part of the study, but especially that injection. We understand that, some people would rather have someone else do the injection so they don't have to look at it themselves. Um, but I will say throughout my long COVID journey, I have had to do many injections when I hadn't ever done it before. And it's not as hard as I thought it would be, or
Hmm.
as creepy is the only word I can think of. Disturbing. It's not as difficult as I thought it would be in any way. Once you get past the first one, I think it's pretty easy. But that's
Right.
obviously going to vary by person. And then there was one more thing I was going to say about the remote participation. Oh, just anytime, I think I said anytime you have a side effect, um, we will have a clinician review your information and think about what we want to do, if anything, what we want to recommend. And then we also have a data safety monitoring board, which is standard for this type of trial, where they are an independent board that is reviewing the data on a regular basis to make sure that there is not undue risk for participants that was unexpected.
Hmm.
So if we see, for example, a very high rate of severe adverse events, that are causing people to be hospitalized at a much higher rate than we had seen for this drug before, the Data Safety Monitoring Board can make the choice that the trial needs to stop because of undue safety or the dosing needs to change or whatever. They can make that sort of recommendation.
Hmm.
Also, at the 3 month point, the Data Safety Monitoring Board will get the chance to look at unblinded data and make the choice of whether, they want to stop the trial, of course, or if stop it for negative effects or stop it for positive effects. So if the drug is just doing amazing things and they feel like, wow, it's really not ethical to have people on placebo, everybody with long COVID should get this drug, they can make the choice to stop early. So that is made by an independent group that's not, as involved with the outcome of the study as the study team is. I want to emphasize for people that's very unlikely, but I also want them to know that we're looking for that. We're thinking about the possibility that if things are great, we might not want this to drag on for a whole year.
Right. Right. Yeah, I was thinking that too. And it's so interesting because as you're talking about these things, I think about themes like healthy aging and aging
Yes.
in place, which has been such an important topic for our show. And clearly, we've had the obviously the sequential development of the GLP-1s, right? Diabetes and weight loss and cardio, and now this Alzheimer's stuff that's happening, which is
Hmm.
awesome. what you're doing is sort of, again, in looking at a syndrome like this, and most people think something like aging as well. It's not one thing; it's a basket of all these different organ systems and things that ultimately go wrong in terms of loss of resilience over time and all that. Love to just, while we have you today, Julie, just your thoughts on how these models potentially can be used to do things that maybe, traditionally, a pharmaceutical company might not want to study multiple diseases because it's harder and longer. But under, what you're doing in these really elegant approaches in this concept, you have the ability to do things that, have not been done before, whether they're repurposing, whether they're studying baskets of diseases as opposed to single. Can you just, thoughts in general on this area, and then related to the concept of aging in place and aging in the home?
Yeah, absolutely. It's, this is something that sort of intersects with the way that we think of the All of Us research program, where that was sort of is a taking all comers, approach, where we're just saying, okay, we're going to get representation from all different diseases because we are inviting anybody in. And then in that database, you could say, okay, I'm going to study people with diabetes and then everybody else is my control. I'm going to study people with cancer and then maybe some of the people with diabetes are going to be the control. And in
Right.
that way, you can sort of multiply the benefit of the data.
Yeah.
And so I love things like that where you're looking at different cohorts together. I do think it's amazing how much this drug is doing and how many times in science in general, drugs are repurposed
Right.
away from their initial purpose because either we didn't realize a shared mechanism was there, or we did realize it, but it took some time.
Yeah.
I think this drug really emphasizes something that I have seen over and over in my career, which is that we need to be humble about how much we actually know about how the
Yeah.
human body works, right? There is so much that we don't know. It's like, that analogy about, looking for your keys only where the spotlight is shining. I feel like that's how we are in terms of our understanding of biology. So it doesn't surprise me that this drug is not this drug in particular, but that there is a drug that is doing so many different things and helping with so many different diseases because we don't really know what's going wrong in the body. Um, in terms of the aging in place piece and, remote work in general, remote, not work, but remote studies, I
Yep.
think it's crucial for inclusion for many different reasons. There are a lot of people who have childcare responsibilities or, other
Yep.
reasons that they can't leave the home. But certainly for disabled people, it makes a huge difference to be able to access something from your home versus having to get there for and overcome whatever access challenges you have.
Hmm.
The other thing that I think is really interesting about it is the multilingual component, where we can translate things into any number of languages and be able to reach people that have, maybe limited English proficiency, but certainly understanding in other languages.
Hmm.
So I just think there are so many reasons to take this sort of more broader accessibility and join us from wherever you are type approach. And I hope it does become the industry standard in coming years.
Yeah, absolutely. And then just as a note again for the audience, Lockett is just one thing that you're doing again on the sort of the digital medicine component of the website is lots of, everything from precision nutrition to really interesting sort of opioid reduction technologies where with wearables. Can you just again say a couple words about some of the other stuff
Yeah.
that's going on there, because it really is not that Lockheed isn't cool, but there's a lot of other really cool things
Yeah,
happening
no, absolutely. This
simultaneously.
approach can be applied to so many different things. I'll tell you about another study that I led along with Eric, which was called
Cool.
ImmunoCare, and that was
Okay.
looking at a immunocompromised population and their exposures to COVID. And so,
Yeah.
of course, we know that a lot of people that are immunocompromised cannot mount the response to the virus that an average person might, even with vaccination. A lot of times they could have as many vaccines as they want, but their immune system is not able to, sort of recognize the virus and attack it the way that an average person would. And so they are at much higher risk of a acute COVID infection. And many immunocompromised people, including some that I know, just had felt that they had to take so much more COVID precautions. Even when everyone else is out there doing life as normal, they're still behaving like it's March 2020 because their life depends on it. And so we did a study where we sent half of participants 10 at-home COVID tests per month. And this was through Q Health, who is no longer around, but they were these at-home nucleic acid amplification tests, which is basically PCR equivalent, much more
Sure.
sensitive than a normal rapid test that you do that's a sort of antigen test. And so we sent them to half the people we didn't send them to. And then we compared outcomes in the group. And so what we were really hoping was to decrease the number of COVID infections at first because they were able to share the test with people in their household. So my thought
Hmm.
was, well, if I'm immunocompromised and I'm staying home all the time, but someone else in my household is going out, to the office or the grocery store or whatever, they're going to be my main way of getting the virus. So if they could test early in their infection and isolate from me, that would really help. And then we also wanted to look at the individual themselves, of course, if with the idea being that if they had a very sensitive test that they had access to for free at any time, maybe they could catch the infection in the early stages and then avoid it escalating to the point that they needed to be hospitalized.
Hmm.
So our overall goal was to reduce hospitalization. What we found in the data was that the rate of infection was basically the same. The rate of hospitalization was directionally lower in the people that had the access to the test, but not statistically significantly lower. But the ICU stay rate was much lower. It was like
Hmm.
one ICU stay in the group that had the tests and 11 in the group that did not have the tests.
Hmm.
So that is a major advance for the people that are, of course, experiencing this, but also we were trying to make the argument to payers that it's worth paying for these tests because an ICU stay is very expensive. The other piece of the intervention I should mention is there was same-day telemedicine access and delivery of medication. As long as they had the appointment before 3 p.m., they could get the medication on the same day. So they were able to really act on the knowledge that they had an infection really quickly. So that really suggests for people that are immunocompromised, make sure you keep testing and take the medication that you can as quickly as possible. But that was another one that, immunocompromised people were not going to want to come in to the clinic to be able to, just for a research study,
Right.
right? So to be able to reach them by sending everything to them in the mail was really, really helpful for being able to reach the study goals.
Yeah, and that just again for the audience, that study is published in Mayo Clinic Proceedings, randomized trial of at-home COVID-19 test, telemedicine, and rapid prescription delivery for immunocompromised individuals. And you'll see everything that Dr. Vogel just talked about there.
Yes, we can make sure everybody gets a link to that too.
Yeah, no, we'll put a link to that one as well. Yeah, no, it's really impressive, and again, it's very elegant the way you think through these different models. It's very impressive. Um, Julie, what else should we be on the lookout for? I mean, clearly quite a few programs going on. Anything coming up that's public facing that we should know about as we get into 2026, whether it's Scripps related, I don't know if you're going to be getting out on the conference circuit at all anytime, but other things that I missed while we have you today, please.
Yeah, of course. One of the things that I'm really excited about that's another long COVID study we did that we're analyzing data on now was the long COVID wearables study, where we
Okay.
were using wearables to try to teach people pacing better. So that's this concept
Yeah.
of, if you go over your energy threshold or outside your energy envelope, we call it a lot of times, you can trigger post-exertional malaise or a really out of proportion worsening of symptoms following exertion. And so we distributed wrist-worn wearables to 400 different people. Some people got it on day one, some people got it after 3 months. And then we compared the difference between those who, at the 3 month data point, between those who had had it for 3 months and those who didn't. And we also sent general pacing educational materials. So we are analyzing the data on that now, and I hope to be able to publish that in Q1 2026. It's hard to believe it's been that
Hmm.
many years. And we're also doing some really interesting exploratory analysis on that, looking at the different clusters of severity of long COVID by both survey metrics and by wearable data. So we certainly see that the severity on wearables matches the severity on surveys, although there are some outliers.
Hmm.
For example, there's that idea that, some people have cognitive post-exertional malaise more than physical. So they might be able to go for a hike, but not work on their computer. So they might
Right.
have a relatively severe score on a functional capacity measure, but still a high step count. So there are some outliers like that. But one of the things that we're trying to do through both that study and in Lockit, where we're deploying the wearables to the participants, is explore whether wearable data can replace typical patient-reported outcomes for clinical trials. And there's two reasons for that. One is that the wearables are objective measures. It's just watching what you're doing and reporting on it. It's not subject to recall bias or those sorts of things that some people worry about with patient-reported outcomes. Um, but also, it's what was I going to say was the second thing about it? Oh, lower patient burden, of course.
Sure.
So if you're wearing a wearable, you just put it on and wear it for the day and that's it. Whereas for a survey, every single time you have to go through, click throughout the questions every time we want the data point. And one of the really interesting things we saw in the long COVID wearable study was that there was steeper drop off in terms of people not completing tasks in
Okay.
surveys than wearables.
Got it.
So some people would continue to share wearable data, but not continue to share survey data. And that was more likely to happen in people with more severe long COVID symptoms. So again, it's an accessibility point and a, data missingness point that if you're not collecting data from the people that are most severely affected, you don't know how your trial is affecting them. And there's just a systematic exclusion problem. And so we're really trying to fight against those systematic exclusions.
Gotcha. Gotcha. It's really impressive. It's
Thank you.
again, I'm the elegance of what you've set up and the way you have to think about these different conditions and organizing these trials in an entirely different way than they've been done before. But yeah, it's a whole new era again of digital clinical development, and it's going to be exciting to see where it all ends up. But
Yeah.
you're right in the epicenter of it all, so that's
Yeah. Oh,
that's
one
really
other thing
exciting.
I wanted to tell you about Locket is
Sure.
I just did a check today on how many states we have represented. So we launched just under 2 weeks ago, and we have 46 states in DC we have participants from. So the states we're missing that I want to let people know is
All right.
North Dakota, South
Mm.
Dakota, Mississippi, and then we're also missing Puerto Rico, where we're open to. So anybody in those states, I would love to be able to say we enrolled in every state we were open to. Would love to have those folks enroll, but also anyone with long COVID, of course,
Mm-hmm.
especially if
Mm-hmm.
people can share with those that might not see things on the internet, might not, have as much digital access. I would love to make sure that we're, being as representative as we possibly can.
Yeah. And then this fits nicely again into your paper from in the Lancet in terms of tech-witty and
Yes,
ultimately, using technology to properly reflect the demographic balance
that's
of the
right.
general population. So yeah, I mean, it's again, it's elegant and it fits so nicely together in this story. So again, I'm excited for you and your team, and I mean, obviously, I hope you start doing better and in I don't know if you're even allowed to get maybe the open label part of the study that you're allowed to be in, whatever. But no, it's really great work, Julie, and it's just I look forward to continue to follow you and your team and everything that's happening in 2026 as you continue to develop these novel programs. But really exciting work. Again, for everybody out there that is going to be listening to this episode of the show across the various podcast networks or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Julia Moore Vogel, Assistant Professor, Senior Program Director, Scripps Research Institute. We will put a link to several of the papers that we discussed, but also to the long COVID treatment trial for anyone that wants to check it out or potentially sign up. But again, Julie, I want to thank you for taking the time out of your schedule. I know you're quite busy to educate us, to walk us through everything you've been up to the last several years. Thank you for everything you do for patients. And as again, we like to say on our show, thanks for helping to create a better tomorrow for so many people out there via what you do. Really a great story.
Beautiful. Thanks for having me.
Really great having you.